welcome to Planetterrian Daily, episode 94 for June 19th, 2026. Let's see what the latest research is telling us. Amyloid beta disrupts tau protein function before plaques form in Alzheimer's models. 2 patients remain free of disease activity 15 years after autologous stem cell transplants for neuromyelitis optica. The therapy resets immune tolerance by depleting autoreactive cells rather than targeting inflammation directly. This represents the longest reported follow-up for the approach in any antibody-mediated neurologic disease. The transplants use the patient's own stored precursor cells to repopulate the immune system after depletion. Larger trials are now assessing safety across broader patient groups, from durable immune reset in adults to how early life experiences can reprogram cellular machinery decades later. Childhood abuse or neglect links to elevated mitochondrial respiratory capacity in adulthood. The adaptation ties to cellular stress responses rather than simple energy deficits. Researchers propose this mitochondrial shift may contribute to later physical and mental health differences observed in affected populations. Analysis connected the early adversity measures to the persistent capacity changes through longitudinal health records. Further longitudinal studies are needed to determine whether the mitochondrial shift is reversible. Researchers mapped reefs that maintain function under elevated temperatures across multiple ocean basins. These sites show lower bleaching rates during recent marine heat waves compared with surrounding areas. The work suggests larvae from these strongholds could seed recovery in degraded zones if protected from other stressors. Next steps include field trials to test larval dispersal models under continued warming scenarios. Source: arstechnica.com. A photonic platform at Southern University of Science and Technology captured 5 distinct phases of localization physics simultaneously. The setup used controlled disorder to track transitions that theorists had predicted but not previously measured together. Results indicate localization behavior is more varied than earlier models assumed. The team plans extensions to other quantum platforms to test generality. Source: phys.org. The chosen concept will examine how solar activity and atmospheric dynamics alter the near-Earth space environment. Data are expected to improve forecasts for GPS accuracy and satellite drag in low Earth orbit. Instruments will focus on energy transfer between the upper atmosphere and magnetosphere. Launch timing remains under review pending full mission development. Source: phys.org. Laboratory work on meteorites has validated protocols the Rosalind Franklin rover will use to detect organic signatures. The test confirmed that sample handling preserves potential biosignatures under simulated Martian conditions. Researchers adjusted extraction methods to account for perchlorate interference. Results will guide final instrument calibration before the 2030 landing. Source: reddit.com. Expanded cultivation of palm, coconut, and soybean correlates with documented losses in vertebrate and invertebrate diversity. The study quantified habitat conversion across major production regions and compared it against extinction records. Effects extend beyond direct deforestation to include pesticide and water use pressures. Authors call for supply chain mapping that incorporates biodiversity metrics. Source: phys.org. XRI-SM observations of NGC 4151 detected outflows powerful enough to remove molecular gas reservoirs. The winds originate near the central supermassive black hole and extend into the galactic disk. This mechanism offers one explanation for the unexpectedly low stellar mass in some massive ellipticals. Additional targets will be observed to assess how common the process is. Source: ScienceDaily.com. Australian Antarctic program voyages documented virus arrival and mortality patterns on Heard and McDonald Islands. Genetic tracing points to introduction via migratory seabirds rather than direct human contact. The preprint details species-specific death rates and environmental persistence. Publication in a peer-reviewed journal is pending. Source: phys.org. From mitochondrial reprogramming after early stress to another early molecular event, this time in neurodegeneration. Amyloid interferes with tau's role in stabilizing neuronal structure in cellular models. The interaction occurs prior to visible plaque and tangle pathology. Researchers used cellular models to isolate this early molecular step in the disease process. Work now focuses on whether blocking this specific step alters later neurodegeneration. The study shows the interference precedes the classic plaque and tangle pathology visible at later stages. Fathers taking 14 to 40 weeks of leave showed lower rates of depressive symptoms than those limited to 4 weeks or less. The association held after controlling for income and pre-existing mental health. Financial barriers were the most common reason internally discharged, examining whether policy changes that increase access produce measurable population-level effects. Source, reddit.com. From protein-level disruption inside neurons to an enzyme that modifies its own activity outside the cell. A Nagoya University team shows a single enzyme performs synthesis and reactivates after chain cleavage. The cycle overturns the assumption that separate enzymes are required for polysialic acid addition and removal. The finding clarifies regulation of the modification essential for neural development. Researchers tracked the enzyme through its exit from the cell and subsequent reactivation after cleavage. Follow-up work will test the mechanism in additional cell types. Standard genome reconstruction methods produced inconsistent ancestral states when applied to large modern datasets. The Canadian study demonstrates that increased data volume can amplify errors in phylogenetic inference without improved models. Revised trees alter estimates of when key metabolic pathways emerged. The team released updated algorithms for community use. Source, phys.org. From fundamental cellular mechanisms to a practical biotech application of precise gene addition, 2 studies achieve efficient, site-specific DNA addition at the 25S ribosomal locus in tobacco and rice. The method avoids random integration risks of earlier transgenic techniques. Both studies report stable inheritance across generations without disrupting essential gene function. The insertions targeted the ribosomal DNA locus specifically in each crop species tested. Field testing in additional crop species is planned. From engineered precision implants to persistent mismatches between marketed and actual microbial content in supplements. Analysis of 352 supplements found microbial content often unrelated to marketed effects. University of Virginia researchers built computer models from the data that predict which strains influence specific functions. The work highlights gaps in current labeling requirements. The models aim to guide formulation of more targeted products in future work. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And before you go, this show is part of the NARA Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give Unintended Consequences a listen. True stories of good intentions that backfired. You can explore the whole lineup at neuranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.